My name is Patrick Arbuthnot, and I'm the Director of the Antiviral Gene Therapy Research Unit, which is also an extramural unit of the South African Medical Research Council. So we, together with the team of synthetic organic chemists on main campus at Wits, a team of Professor Charles de Koning, looked at trying to develop a green method for making these ionisable lipids. So the traditional way of making ionisable lipids is that petroleum products are used to generate the precursors or the feedstock, the basic building blocks that are used to make ionisable lipids. So it was felt that if we could use something that was green and also abundant in Africa, that would be quite a good thing to enable us to have freedom to operate and not be dependent on patents of the so-called global north. So we set about this project with the team of synthetic organic chemists, and they took material from cashew nut shell liquid and they made ionisable lipids using these compounds which they extracted from the cashew nut liquid.
Hi, I'm Charles de Koning, Professor of Organic Chemistry, full professor since 2005, so that's 20 years. I'm actually a University of Cape Town graduate from 1988. Then I went to MIT to do a postdoc, then to University of Hawaii, and I'm what's called a synthetic organic chemist, and I've been doing this for, I don't know, 35 years or something like that. We really started this in earnest in April 2022. The project was funded by the World Health Organization and the Medicines Patent Pool. And now we're 3.5 years later, and we do have lots of, in fact, we've synthesized more than 100 ionizable lipids, and these are much cheaper than the commercial ionizable lipids that were used in South Africa, for example, in the Pfizer-BioNTech vaccine, or in other parts of the world, the Moderna-based messenger RNA vaccine, where the ionizable lipid in that particular case is known as SM102. Currently what we're doing is we've made about 100 different ionizable lipids, and we are checking whether they're able to deliver messenger RNA and express, just like the COVID vaccines, express the spike protein of COVID, and we've had quite a lot of success in this.
Hello, my name is Robin Clintworth. I'm a synthetic organic chemist working at Wits University. I got my degrees here, and I also did my PhD here at Wits University. We're working on the synthesis of new ionizable lipids from biorenewable waste as a precursor chemical, specifically cashew nut shells. So it's the inedible shell that surrounds the nut. The nuts are obviously consumed by people all over the world, and for every nut you have a shell that just gets discarded. And those shells are incredibly rich in phenolic lipids, which turn out to be ideal precursors for ionizable lipids, which you can use for mRNA delivery, for the next generation of mRNA vaccines. So the first vaccines that were ever developed essentially used either modified viruses, like smallpox vaccine required the cowpox virus, and that actually doesn't infect humans, but it has enough of the same similarities molecularly to generate an immune response.
I'm Connor Stockley. I'm a third-year PhD student at the University of Witwatersrand, and my PhD is focusing on synthetic organic chemistry, and our team is aiming to address the issue of vaccine inequity in Africa, and this comes down to particularly two main markets that we are focusing on. The first one being that of transfection agents, and whilst the second one is that of raw materials for mRNA vaccines. Now, transfection agents is a large market, having about a market size of $1.3 billion. And most research facilities around the world are heavily dependent on these transfection agents. However, access to these is extremely expensive, and this severely hinders research and slows down academic research. Then for lipid nanoparticles, the raw materials for these, particularly ionizable lipids, are extremely expensive and are covered quite severely by intellectual property. And this leads to Africa only producing about 1% of the local vaccines that are distributed within the continent. And so this opens up a huge market for growing vaccine development within Africa. And there's been a strong push to increase vaccine production in Africa from that 1% up to 60% by 2040. Now, our lipids are produced from cashew nut shell waste, and this again addresses the issue of local manufacturing, as we can obtain this from African sources, and this limits the amount of raw materials that need to be imported from overseas countries, and that majorly reduces the cost. And in doing so, hopefully we can find that gap in the market and address the inequity within African vaccine development.